We get started, just a reminder to please enter your questions at the end of the presentation. Please enter them in the chat, and please answer a brief survey at the end of the presentation, and that will let us know that you were present today. We have a special speaker guest lecturer today that will be introduced by Dr. George Fantis. This is the McGill Annual Novo Nordisk Lecturer. So very special lecture today. And George, I will ask you to please introduce our guest speaker. Thank you.
Okay, thanks, Nadia. So it's a pleasure for me on behalf of the Division of Endocrinology to welcome Dr. Doug Melton today as the McGill's 38th Novo Nordisk lectureship holder. So this lecture has been going on for a long time. And before I go on to introduce Dr. Melton, I'd like to thank Novo Nordisk for long-standing support.
Fanor, s'il vous plaît.
Pardon?
Abraham Fanor.
Oh, I think someone should be muting themselves. So thank you, Novo Nordisk, for long-standing support for this lectureship. So I put on a little slide here to advertise that Dr. Melton is giving rounds, but also we'll have a more detailed scientific lecture at, actually it's at 5 p.m. Not, there's something, this was made incorrectly. It's 5 p.m. And the Zoom link is there for those of you who are interested. So Dr. Melton is professor of molecular and cellular biology at Harvard University. He holds 2 chairs, the Thomas Dudley Cabot Professorship in Natural Sciences and the Zander University Professorship. He's co-director of the Harvard Stem Cell Institute and an investigator of the Howard Hughes Medical Institute. He had a broad undergraduate education in both biology and the history and philosophy of science, and then did his PhD at Cambridge in the UK. He began at Harvard in about 1981 and worked on regulation of gene transcription. In the 90s, he moved on to embryogenesis and development in mice and worked on the ectoderm and the pancreas. And he's really developed really world-leading, outstanding, high-impact papers discovering how the beta cell, the insulin-secreting pancreatic islets, develop from embryogenesis to mature islets. So in the area of regenerative medicine and stem cell research. He's won, of course, many awards. He's been inducted into the National Academy of Sciences, the American Academy of Arts and Sciences, the Institute of Medicine, and the Japanese Biochemical Society. He's won the Kayla Grotsky Basic Science Research Award, the Juvenile Diabetes Research Foundation David Rumbaugh Award of Scientific Excellence, and the Ogawa Yamanaka Stem Cell Prize, along with the Elliot P. Joslin Medal for Diabetes Research. He served as president of the International Society for Stem Cell Research and has many honorary degrees and sits on many committees all over the world as an authority in stem cell biology. So the Division of Endocrinology and Metabolism at McGill is honored and delighted to welcome Dr. Melton as the 2022 Novo Nordisk Lecturer. So Dr. Melton, I'm gonna stop sharing and can put on your slides.
Wonderful. Can you now see my slide, George?
Yes.
Yeah, great. Well, thank you for that introduction. And more importantly, thank you for the nice invitation to join you all. As I'm sure everyone would agree, it's nicer to do this in person, but we're all getting used to Zoom life. And so I'll do my best to make this interesting on Zoom. The first slide here says what I'd like to talk about today, which is using human stem cells, embryonic stem cells, to make islets for the treatment of diabetes. And I'll talk about a number of things, but I thought I would start by just saying, what is the sort of goal of my lab? It's simply stated to try to cure type 1 diabetes. Now that will make many medical doctors smile because curing something is not trivial. Usually it can't be done, we treat it. but I don't see any reason not to aim for a cure. And so by way of setting the stage, I'm not so interested in working on preventing the disease or studying its complications or doing anything about policy. I'm really just focused on the idea of trying to change the course of treatment for a patient by curing their disease by transplanting beta cells. So I'm going to take you through a sort of personal perspective on why I think that's possible. The first part of my talk will summarize some published work and end with the results of the first clinical trial. But then I'm going to spend a little time at the end showing you or talking about how a developmental biologist thinks about improving on this and where I think the future could go. And I'll try to finish in about 45 minutes or so that we have time for questions. But I realize I want to go back one slide to point out something important here at the bottom. I'm the founder of a company called Sema Therapeutics, which has been acquired by Vertex. And I'm going to talk about some results at Vertex, but that's my disclosure as a conflict of interest. Okay. This is how I think about the problem. This will be known to many of you, which is that pancreatic islets, the number for which is about 500,000 of these are found in the human pancreas, has 5 cell types that make 5 different kinds of hormones. And the important one, well, the most important one is insulin, shown here in the blue staining cells. This is a confocal image of one pancreatic islet, which contains typically about 5,000 cells. Some other cells here not highlighted in color, but in green, say, would be the glucagon-producing cells and the other 3 there. And as you will appreciate in diabetes, the insulin-producing cells are either lost in the case of type 1 diabetes by autoimmune rejection, or in the case of about 10% of type 2 diabetics, by stress or overuse, they become dysfunctional. It's wonderful to talk to people in Canada because no nation has made greater advances on this subject of treatment diabetes than Canada. One will, of course, note recently there's been the celebration of the discovery of insulin almost exactly 100 years ago by Banting and Best. And one of the things I like to show undergraduates here is this picture. That is, for insulin-dependent diabetics, the treatment has been insulin by injection for nearly 100 years now, more than 100 years. And I like this picture. I'm not exactly sure where it's from, but it's a picture of 2 tons of pig parts from which about 8 ounces of pure pork insulin could be biochemically isolated. And that was the way diabetics were treated in the early part of the last century, first with pig, then with sheep, then with cow, and other sorts of insulins, all purified based on the work of Banting and Best and their colleagues. In a sort of perhaps unfair way, one could say there's not been a lot of progress. There's been the cloning of the human insulins, and then variations in the structure of the insulin to make short and long-acting insulins. Novo Nordisk has been active in this area. Their introduction of the pen in '96 was quite important. Then Sanofi's long-acting insulins, et cetera. But I think it's fair to say, as shown here in this picture, that for now nearly 100 years, there have been improvements on injecting insulin and using monitors, all trying to substitute for the absence of the beta cell. Among the most impressive advances, I would say, on the technological side, beyond the creation of modified insulins, has been the use of pumps. Here I'm showing you a picture of the diabetic, or let's say the blood glucose monitoring of people with and without diabetes. In the top picture, you can see in the green range from 70 to 120 milligrams per deciliter, a normal person's blood glucose over a 24-hour period. A very typical person using a pump is shown in the middle, and using a dual hormone pump developed by Ed Damiano and their colleagues and others, you see at the bottom more time in the, quote, good region in the green range. But still there are excursions due to meals, which take the blood glucose out of normal range. And as many would appreciate, it's the high blood sugars which lead principally to the complications of peripheral neuropathy, cardiomyopathies, and other problems due to having diabetes. I don't in any way want to demean or belittle the enormous advances and investments that have been made in treating diabetes with dual hormone pumps and improved insulins. But I would nevertheless like to contrast this with work that was begun by Paul Lacey and his colleagues, as far as I know, in Washington University about 50 years ago, and then developed to a kind of fine art by James Shapiro and Ray Rogette and their colleagues in Edmonton, which is the transplantation of islets. So here's a picture I often have in my mind. This was given to me by a colleague, Gordon Weir. These are human cadaveric islets ready for transplantation, although in this case, they've been stained with dithazone, the stain for insulin. So these are, as you may well know, isolated from cadavers by enzymatic digestion, and then transplanted into the portal vein along with non-steroidal immunosuppressants. And this is a picture that I think is important to focus in on, is at the top are the tracings for about 3 or 4 different diabetics, type 1 diabetics, looking at their blood sugars before islet transplantation, and in the bottom, their blood sugars after islet transplantation, along with immunosuppression. So let me pause there in a kind of superficial way by saying that because of the work done in the 1920s on the discovery of insulin, and then on the advances by others, Shapiro and colleagues and Lacey and colleagues on islet transplantation, it seemed clear to me that it would be possible to treat the disease more permanently, what I would call a kind of a cure, if one could make more beta cells and deal with the immune system. So that's what I'm going to spend my time on today. First, talk about the loss of beta cells and whether or not one can just make enough beta cells to treat patients. And then I'll move to how to manipulate the immune attack to try to induce tolerance to the stem cell-derived beta cells. This is work I began in my lab, nearly 25 years ago. And the first question was how to make more beta cells. So I'm showing you here the 4 different ways we tried, and I'm going to focus on the last one, but just say a bit about the other 3. So when we started this work, it was believed that you could stimulate endogenous stem cells that might be found in the pancreas, that could be caused to differentiate into beta cells. We spent some time studying that and concluded that there were no adult stem cells. That is, there were no stem cells in an adult which could be stimulated to make new beta cells. Others have claimed this is true. I don't find those claims very compelling. And certainly, even if their results were right, they make such a trivial amount of beta cells that they would be physiologically irrelevant. We then moved on to try to replicate pre-existing beta cells because we had shown that new beta cells come from old beta cells. But again, we weren't able to show that this could be done at a level that would make a difference to a patient. We also tried what I call direct reprogramming or transdifferentiation. That was turning one kind of pancreatic cell into another. And here we showed by transfection with 3 transcription factors, we could turn exocrine cells in the pancreas into beta cells. But in the end, we decided that there was no easy way to do that in patients. And again, the numbers of cells we could get were not sufficient. So now to what did work, which is to direct the differentiation of embryonic or induced pluripotent stem cells in vitro to form functional beta cells. I'll go over this rather quickly with just a few high points, but I want to say, particularly for those who don't work in the field, what was involved and how it was done. So this next picture is a picture that my lab makes fun of. They call it my Skittle diagram, but it's a picture of turning embryonic stem cells shown on the left as ES cells into definitive endoderm, then the pancreatic gut tube, then pancreatic progenitors 1 and 2, then endocrine cells, and then beta cells. And when I began this work, I naively thought, or maybe arrogantly thought, this would take 4 or 5 years. It actually took closer to 15 years to figure out how to tell a stem cell, which can make any cell in the body, become a functional beta cell. What is shown in blue are that we added different kinds of factors, and I'll show you those. Well, maybe I won't even show you that. They're published. At different kinds of factors for different lengths of time. And this turned out to be about a 40-day procedure. The factors we added are small molecules like retinoic acid and known growth factors like keratinocyte growth factor, or we would block Wnt signaling with a Wnt agonist, et cetera. So we didn't discover any new factors. What we did is figure out which factors to add at what concentration and for how long. And what you see at the bottom are cadaveric organ donor islets, and they're stained for two things. They're stained for a transcription factor in the nucleus, this transcription factor called pancreatic duodenal homeobox number 1, known to be important for beta cells, and stained in the cytoplasm for insulin. So this is a cadaveric islet, which I showed you at the beginning. And over on the right are our stem cell derived islets. And here you see beta cells, which look at this level of resolution, indistinguishable from those of the cadaveric islet. Now I'll show you some molecular data to prove that these are beta cells, but my favorite sort of example or assessment is shown here. This is kind of old-fashioned transmission electron microscopy. And if you look in the top panel, you see a human beta cell isolated from a cadaver. And if you remember from your endocrine histology, here is a vacuole inside the cell with crystallized insulin inside. And when the cell receives high glucose, these vacuoles fuse with the plasma membrane, releasing insulin in the blood to control blood sugars. At the bottom is our stem cell derived beta cell, which has an indistinguishable vacuole and crystallized insulin. So the conclusion here is that stem cell derived beta cells look like functional beta cells. And my point would be, you can't turn an embryonic stem cell into a cell that looks like that, with all the mitochondria, all the vacuoles, et cetera, unless you have turned on and off all the right genes. And I'll come back to the gene expression. But let's just say there are about 3,000 genes that have to be turned off, and another 2,500 that have to be turned on in the right order to make these functional beta cells. What's more important in a way is whether they function. And here I show you a picture to demonstrate function. This is an in vitro assay where we look at the new differentiation here on the left, the stem cell derived beta cells, and we challenge them with glucose. You can think of this like breakfast, lunch, and dinner. There's low and high glucose, low and high glucose, low and high glucose. And in each case, the cells respond to the sequential glucose challenge by secreting the correct amount of insulin measured here in international units per cell. On the far right are adult or cadaveric beta cells. Again, you see low and high glucose, low and high, low and high. And then depolarization in both cases with potassium chloride to release the insulin found inside the cell. In the middle panel are pancreatic progenitors like those devised by another company, by ViaCyte, which they've put into patients, and I'll say a bit more about that later. Those cells don't function in vitro, and they can produce some functional cells in vivo after now has been demonstrated 1 year in humans. Let's focus on the ones that work. And I want to show you a bit more about them, about what they look like, and then about what use they've been put. So here's a picture of these stem cell clusters that contain beta cells. And I want to make clear that I've implied wrongly that when we do this procedure, all of the cells become beta cells. That's not true. In this case, which is sort of typical in our hands and not a great production, about 16% of them become beta cells. Not shown here are about another 20% that become alpha cells, or 15%. And then there are some other cell types, and I'll come back to that later. Nevertheless, these cells, these clusters, let's call them stem cell derived islet clusters, are able to cure or to control blood sugars in 4 mouse models of diabetes, in a rat model, in pigs, and in monkeys. And that was done by us with our collaborators over the last 4 or 5 years. But about 4 or 5 years ago, maybe a little more recently, I decided it was much more important to get these into humans than to continue to prove their function in animals. So I'm now going to talk about work not done in my lab, but done at Vertex. And I'll remind you that I'm a paid consultant for Vertex. So I thought it'd be interesting to see what these cells look like. Here's a little vial where we've taken these human islets made from embryonic stem cells. They've been stained dark blue for insulin using a chemical stain. And each of the dots here contains about 5 to 10,000 cells. But the point of this little vial is to show you how many cells you need for a curative dose. In some ways, you could say it's pretty amazing that that kind of milliliter or so of cells is responsible for this giant market, if you see it from a business perspective, of selling insulin. I read in a newspaper recently that the insulin market, counting the pumps and the monitors and the sale of insulin, this year will be $37 billion. And in a way, that's all the result of the absence of those cells I'm showing you there in that vial. So let's think about it this way. Vertex had acquired this company, Semma. Semma had improved on the protocol that we had made, done it with clinically compliant cells, and then figured out how to scale the manufacturing, all stuff which I did not do. And they've gone into a clinical trial. Some of you may have heard about the first results reported last autumn. This is by transplanting these stem cell derived clusters through the portal vein into the liver, along with non-steroidal immunosuppressants, a variation of the protocol, which is commonly called the Edmonton protocol. And here I just show you these results, again, emphasizing these are results from Vertex. So in the first patient, had been requiring insulin for more than 40 years, complained rightly of constant monitoring of his blood sugar and insulin. He'd had 5 severe hypoglycemic events in the previous year. And importantly, he had no detectable insulin production. The stem cell derived islets, which Vertex calls VX880, were infused into the liver along with immunosuppressants. And afterward, he said, this has changed his life. He makes his own insulin. He's making therapeutic levels of insulin. Now it's, I think, closer to 9 months with only a half of what we calculated at the dose. This is from the Vertex press release. And I want to make a couple of points here. The first, that he had undetectable fasting C-peptide or insulin levels, and undetected even after he was stimulated with a mixed meal tolerance test. This was his hemoglobin A1C measure and his daily insulin dose. And after what we calculated to be half a dose, just 6 months later, he was 90% off of insulin. He reports now that many days he's not taking any insulin. And so let me pause here by saying a few things. First of all, I want to emphasize I'm not a clinician, a medical doctor. So for me, this was quite exciting as a research scientist to see that things we'd done in the lab actually could help a patient. Secondly, let's be clear and emphasize this is one patient before I get carried away. There's an ongoing clinical trial. It will require more patients before secure or firm conclusions can be drawn. But from my perspective, this confirms what we had shown in various animal models, namely that these cells work. So we can talk in the discussion about whether one should get overly excited about one patient, or there's a few more patients now being treated. But I'm going to just assume that it works in people and look to the future. So with your permission, I'll say stem cell derived islets derived from human embryonic stem cells, they work. So what's next? I want to now look to the future to talk about gaining complete mastery over the cell composition and eliminate the need for these non-steroidal immunosuppressants. Both of these are kind of tall orders, but as you can probably already tell, I like a big challenge, and I think it'll be fun to pursue these. So let me tell you about what we've done, where we're failing, and why I think success is still possible. The first thing we did, speaking to clinicians, is something you could never do with human material. That is, we looked at every gene and every chromatin change that goes on and off during the development of these stem cell-derived islets by using the process in vitro and isolating cells and looking at, as shown here for chromatin changes, histone modifications. We used what's called ATAC-seq to look at open and closed chromatin. We looked at DNA methylation at every locus in the genome. We then did single cell sequencing. And again, I just point out, this is something you could never do because you couldn't get enough human fetal material, let alone get permission to, should one ever even ask for permission, to biopsy human fetuses, to say what genes and chromatin changes occur at every stage of development, from a pluripotent stem cell to an adult alpha and beta cell. And we learned a lot from this. One of the things I'll show you we learned here is what's in the final composition, what we call stage 6. So the first thing I'll show you, if my pointer works, and if it doesn't, I'll just use colors, is the cells we were aiming for all along are these stem cell-derived beta cells in purple, SC beta cells. We're also very pleased to see, and not so surprised to see, these red cells, which are stem cell-derived alpha cells. A big surprise, well, another thing we weren't surprised were to see non-endocrine cells in some progenitors here in green and brown. But a big surprise are these blue cells, enterochromaffin cells, cells that make serotonin. These cells don't normally exist in the pancreas. There are reports, convincing reports, that they exist during fetal development, but normally they're found in the gut. So they're very like beta cells. They contain many of the same genes that beta cells do, but they're not interesting for us. We want to get rid of them, and I'll come back to that. So in addition to learning all the genes that come on and go off, we learned proteins that are expressed in the membranes of the cells. And so we could use those, for example, as shown here. We use antibody precipitation, let's say. We use iron particles attached to antibodies and then magnetic bead sorting, so that we can make what you see here on the left is a kind of ultra pure stem cell beta cluster, something that doesn't exist in normal development or in vivo, but nearly every cell there is a beta cell. And on the right, you see nearly every cell is an alpha cell. So one of the things we're working on now is to ask, using animal models, what's the right proportion of beta and alpha cells if we want to make a perfect islet for transplantation? Some people in the audience may have an opinion on that, which I'd be glad to hear. I can say that looking at the literature on cadaveric islets, it's very hard to determine what would be the ideal islet beta alpha composition. Anyway, how are we going to get control of that? I'm going to talk to you now about a program, a project, which I would describe in an overview as a developmental biologist's view of how to gain control over a drug product. So this should make some of you smile, because who knows if this will work, but I'm going to tell you how we're going to go about it. This is what I want to do. I want to get control of the composition so that we make a defined number of beta cells, which are the insulin-producing cells, alpha cells, which make glucagon, and delta cells, which I haven't talked about yet, that make somatostatin. And the reason we want to make those three is there's convincing evidence from others in the mouse that if you knock out the somatostatin-producing cells, the alpha and the beta cells don't work as well as they otherwise would. I'm going to maybe amusingly say I don't care about the epsilon or the gamma cells that make ghrelin and pancreatic polypeptide, because as far as I know, diabetics don't have any trouble with appetite, and no one seems to know much. Certainly, I don't know much about what pancreatic polypeptide does. So we're really going to just focus on the first three. How are we going to do that? The idea here is to use the advances made by others using CRISPR genetic modification to gain complete control of the final composition. So we're going to start with embryonic stem cells and then ask, can we create tailored lines which restrict the differentiation potential via genetic editing? So one way you might think about that is if a cell is like walking down a corridor and it has all these open doors, it can become a mesodermal cell and make blood or muscle, or it can become an ectoderm cell and make nerve or skin, or it can become endoderm. And then it has to decide, do I want to be what part of the gut tube? Esophagus, liver, stomach, pancreatic, intestine. If I become pancreatic, do I want to become endocrine, etc. Secondly, which will make immunologists smile because they tell me we'll never succeed at this, is can we add or remove genes that allow us to make a cell that would not be rejected by the immune system? So I'm going to talk about how we do that and just give you a taste of the result. Maybe more than telling you a taste, I'll tell you now we've not solved the problem, but I'm encouraged by what we're able to do. So this is how we do it. This is very detailed slide, but some of you may be interested just hear about it. On the top is shown a map of a virus, which we use to infect the cells, and that brings in an enzyme you'll have heard of CRISPR-Cas9, which can cut DNA. And if it has the appropriate guide RNAs, it will make a genetic modification in the cell. So again, to step back, let's be clear. We're talking about no longer trying to make a normal cell, but make a genetically modified cell, a cell never found in nature that would have special properties. So in our primary screen, you might remember there are about 20,000 human genes, shown here as 19,000. We use 77,000 different guide RNAs using 4 guides per gene. We used 1,000 non-targeting controls. We did this 3 times, and we identified about 800 top candidates that we thought were worth further investigation. We've then done 18 more differentiations targeting these 800 genes. We did this 18 times so we could get statistically significant results. And we've identified about 200 genetic modifications that point us in the direction that I want to go. I show you this partly because if there's some young people in the audience that want to do this, we've worked out the methods for doing this. We could now control the differentiation or let's say slant the differentiation into any cell type. And this took us about 2 or 3 years to figure out how to do this at the right multiplicity of infection with a lentivirus, how to develop FAC sorting assays, how to isolate the DNA from the differentiated cells so they still had their guide DNA so we would know which gene had been knocked out, etc. What we also want to do, though, is not just control the differentiation in terms of composition, as shown here to make alpha, beta, and delta cells, but also make them immune protection. So I'm going to do that again because I'm so proud that I figured out how to make these little Marvel shields. Yeah, so that's telling the immune system, go away, you can't bother me. You either can't recognize me, or even if you can, I'm going to induce tolerance. I want to say a bit about this program because I work in a city that claims to have a lot of experts on immunology. I shouldn't say claim to, it does. And almost every immunologist I talk to about this smiles and says, this is a fool's mission. It will never work because the immune system has so many ways of detecting unwanted cells that you'll never succeed. And to that, I would say it makes science no fun if you think you can't do something. But also, I was told similar things when I said we're going to try to turn an embryonic stem cell into a functional beta cell. So I'm not saying we'll be successful, but I'm all in on trying to solve this problem. So let's talk a bit about immune protection. There are 2 ways one could think about this. I'm going to talk to you about genetic modification, but I'll just quickly summarize what we do with some collaborators and what others are doing effectively is what I would call physical protection or encapsulation. So here on the bottom is a picture of some stem cell derived islets inside alginate beads. And that's one way you could think about protecting the cells. We do this with lots of collaborators. I show you in this slide on the top left, an alginate bead with an islet. So this is not a cell with a nucleus. This is an alginate bead with a 10,000 cell islet inside. And here you can see the results on the right from our colleagues. This case was with Mark, with Dan Anderson at MIT, whereby putting into mice, fully immune competent mice that have been treated with streptozotocin, we can control their blood sugars for more than a year now by putting these alginate coated beads in. I think for bioengineers, this remains a big challenge because the volume of alginate you would have to put in is so enormous, it's not clinically sensible at the moment. But I do think that that approach of physical protection is one worth pursuing. I'm just not doing it myself. So I'm going to take a chance here at the end on diving into the genetic modification for immune protection. And for those of you who don't know immunology, I'm in your camp. I'm just a student learning immunology. So I'm going to summarize what I think is the problem and how we're going about it. So let's call this the challenge of allo rejection. This is the same challenge that organ transplants face, but there are a couple of differences. The first difference here is that the product we would be putting into the patient would be the same in every case. So it's not isolating cadaveric islets. It's making a defined product, the same product every time. Secondly, this will be an allo or non-self islet cell, but in this case, I'll just focus in on the beta cell. Just to remind everyone, this would be an allo cell if we were treating type 2 insulin-dependent diabetics, and it will be an allo cell if we're treating a type 1 autoimmune diabetic, because in both cases, there's going to be an immune reaction. It doesn't do any good to create a type 1 diabetic's own beta cell because they have autoimmunity. So I'm really focused on this allo rejection. What is known from immunology to reject these cells? Well, lots of things. First of all, there's killing by CD8 T cells. One way one might get around that is to remove beta-2 microglobulins so the beta cell can't present its epitope. However, it's well known if you do that, then natural killer cells say, aha, you don't have any class 1 or class 2, I'm going to kill you anyway. There are ways you might think about preventing that. For example, adding inhibitory ligands like HLA-E or G or CD47. Then there's the complement factors. That's another problem to deal with. On top of that, there's macrophages, which are going to find you and eat the beta cell. And then there's activated lymphocytes that are secreting cytokines. So God knows this is a non-trivial problem. Also, that's clear from this kind of undergraduate textbook picture that one is never going to find a gene that will prevent rejection. So we could lower our sights a bit and say, well, can we do enough of these modifications so that while we might not completely prevent the requirement for immunosuppression, could you make the cells slightly opaque so they'll evade the immune system, or that you need rather low doses of immunosuppression? I'm impressed by the fact, which I've just read about, that some non-trivial number of people who receive liver transplants go off immunosuppression. And no one really understands why they then accept this foreign liver. I'll also remind us all that during pregnancy, the mother accepts the fetus when she shouldn't because it's expressing sperm-derived antigens. So in biology, there are ways that the immune system leaves some cells alone. That's just what I'm trying to mimic or copy, appropriate. So what are we doing? In terms of making genetically engineered stem cell derived islets, we are knocking out all of the genes shown here and have already done so. And I would say these are done, I would say this is what a smart undergraduate would do by reading the literature. So let me draw your attention, for example, to from the work in cancer, it's really well known, that PD-L1 and CD47 are quite important as don't eat me, leave me alone signals. Others have shown renalase reduces stress. So none of these are things we discovered. These are just, I'd say, like rifle shots taken from the literature. But I also described for you this here at the bottom by doing CRISPR screening. So I'm going to finish by telling you how we do our CRISPR screening and show you one result and tell you why I think that's hopeful, but that we're not there yet. Okay, what do we do? This is the model we're using, which is a stem cell derived islet allograft into a humanized mouse. So the humanized mouse is a NOD-SCID IL-2R gamma knockout, which allows us to put human cells in that are not rejected. But after about 10 weeks, because we're going to inject human peripheral blood mononucleocytes, there's a graft versus host reaction. So you have about 10 weeks to do this experiment. So we take a humanized mouse and we inject the stem cell derived islets I told you about. We then inject human peripheral blood mononucleocytes. I'll show you experiments where we do it from 2 different donors. So these are allo PBMCs that are going in to attack the human islets in a mouse that, let's just call the mouse like a living test tube. Here's sort of what it looks like. Here's a kidney graft done in the control with no PBMCs, and I hope you can see my pointer. These are the stem cell derived islets in the kidney capsule. Then 10 weeks later, after injecting human PBMCs, the graft is gone. Looking at that in a more analytic sense, look up here at the top. I didn't tell you that we put a luciferase reporter into the islet so we can do AVIS imaging. Looking at luciferase now. Luciferase is driven by a generic promoter from GAPDH. In the control, you see here, 1 week later are the islets that are put in, and here 8 weeks later are still the islets, healthy and happy. If you look when we inject human mononucleocytes, PBMCs, in the control, in the experimental 1 week, and then at 8 weeks, they're mostly gone. It's interesting to look over here. When we use the control of stem cell derived islets without PBMCs, you see insulin production. This is 10 weeks after, and we've injected PBMCs. You see the insulin production goes away. Over on the far right, we measure the number of alpha or beta cells, and in both cases, you see they're being eliminated by the human PBMCs. So I'm going to show you one good result, but before I show it to you, I want to make clear, I do not contend that we've solved the problem. This just is a way forward on the problem. This is a kind of a complicated experiment. We take human embryonic stem cells, we knock in a reporter for luciferase, we have them express IL-10, TGF-beta, and a modified interleukin-2, which is known to induce T regulatory cells. We also knock out class 1, all of class 1 A, B, and C, and replace it with HLA-EF because this is what's expressed at the maternal fetal interface. So this is clearly a weird cell. This is not a cell you would find in nature. The first thing we did is test whether we could still differentiate them into functional beta cells. And you see here, 2 and 20 millimolar glucose, depolarization with potassium chloride. So they're still functional, making insulin. We're now putting these into not a PBMC model, but a xenorejection model. The mouse is an immune competent mouse, and we're saying, can it reject these cells? So if I draw your attention to the bottom left, on the wild type, that is unmodified stem cell derived islets, you see within 2 weeks, the mouse's immune system has eliminated them. On the top, this weird cell we've made has this silly name 2B10 with all these genetic modifications that survives for 9 weeks. And when I was making this slide, I flipped this, unfortunately. So in the wild type case, if you look for T regulatory cells, you don't find any. If you look for general immune cells, you don't find any. They're all gone. You find very few insulin cells left. But in our genetically modified cells, you find a lot of Tregs and insulin-producing cells still around. So let me emphasize again the limitations of this. This is a xenorejection model. It's not human. It's a human cell. We don't know the answer yet, but we need models to test for genetic modification to protect from the immune system. I've already given this talk enough to know that immunologists, as I've said, think this will never work. And I'm glad to hear comments from others and suggestions. But I want to finish with just a couple slides then with my personal view on the way forward. So I believe that people who now inject insulin will in the future get stem cell derived islets. Let me tell you again why I think that's true. The best continuous glucose monitors claim to read blood glucose every 5 minutes. I actually have looked at the data and I don't think that's true, but let's say it's true. I think it's closer to every 15 minutes. But let's say it's every 5 minutes. I'll remind you that a beta cell reads blood sugars, reads blood glucose every millisecond, 1,000 times a second. So in terms of reading blood sugars, CGMs can't hold a candle to a beta cell. Secondly, beta cells, unlike an insulin pump, don't provide a bolus of insulin. They squirt out tiny amounts of insulin all the time to control blood sugars. So what I think we're going to see is transplantation of stem cell derived islets into the portal vein with immunosuppression following the Edmonton protocol. And I've already told you about the first patient with this. We're going to see more of that this year. And I hope that this will continue to work. Actually, I might also just say a personal note here. When I was first shown the results from the first patient, I didn't even understand what I was seeing because I thought I was looking at the control. His blood sugars were so well controlled. I was hoping there would just be evidence that they worked at all. So I think we're a bit lucky in this first case, because actually in the lab, when the first experiment works, it usually becomes more complicated. Anyway, this clinical trial run by Vertex is ongoing, and I'm very excited and enthusiastic about that. Vertex will also use their proprietary encapsulation device that will go into a clinical trial this year. I didn't show you that device or talk about it, but it's a deceptively simple and very clever device that allows for vascularization and still provides immune protection. And that device has worked extremely well in pigs, and I'm hopeful about humans. What I talked to you about today was trying to make designer islets, naked or genetically modified islets for transplantation. We're a long way from that, but my dream is that when a young person is diagnosed with type 1 diabetes and they and their parents are in the endocrinologist's office, she says to them, you know, this is bad news. You have diabetes and that's no fun. It requires daily continuous glucose monitoring your diet, your exercise, insulin injections. Without what I'm going to tell you about, it's a psychosocial cost on you and the family. And frankly, only people who are diabetic or have a family member really appreciate what it means to be a type 1 insulin-dependent diabetic. But she'll say, I have good news for you. In my freezer, I used to have insulin, and now I have these frozen genetically modified cells, which I'm going to inject under your skin, and they're going to control your blood sugars for the next few years. And when they stop working, come back and I'll give you more. So I hope you're smiling about that. We're nowhere near that, but that's something I think we should aim for. I want to finish with a nice picture post-COVID of the people who do this work with me here at Harvard in my lab. Their names are here on this slide in black. In green and blue are my collaborators. I want to thank the Howard Hughes Medical Institute, the Harvard Stem Cell Institute, the Picower Foundation, the National Institute of Health, and the Juvenile Diabetes Research Foundation. I also mentioned, I want to emphasize again, I'm a paid consultant for Vertex. And I'm going to stop there, having saved time for questions, and hope that it worked on Zoom.
Fantastic. Thank you so much for presenting this transformative research. Really outstanding and exciting work. George, I'll let you take over the questions.
Yes, I was going to thank Doug as well. Fantastic talk and very, very exciting to see this in clinical trials. So the chat is open for questions. We have a couple of questions here. One from David Rosenblatt. Could you start with somatic cells from individual patients like fibroblasts and create iPS cells, then generate the islet cells? would that be a better idea because of autoimmunity?
It's a good question. So let's just think that through. If you take a type 1 diabetic and make their own stem cell and then turn that into a beta cell, let's remember that that's the beta cell their autoimmune system was already recognizing and killing. So for a type 1 diabetic, it will not do any good. On the other hand, your idea would work for type 2 diabetics because they don't have autoimmunity. So far, the markets, the business markets have not shown any interest in that because it's so expensive to make individualized iPS cells. But to just follow up a bit on your question, we have made iPS cells from 3 type 1 diabetics and from 5 non-diabetics and made their islets. And we use those as tests with their blood to ask what cells are killing beta cells in a type 1 diabetic and what cells are attending to beta cells in a non-diabetic. So we're using it for research purposes, but I don't see now that that will be useful as a program to give rise to a medicine.
Thanks. Another question relates to the childhood diabetes. So the question is, will the clinical trials be done in children? And that, I had a question related to that as to the exclusion-inclusion criteria. Right now, I understand it's quite difficult to find those patients because hypoglycemia, severe hypoglycemia is a criteria. What do you think of that? And will they be sort of loosened a little bit to allow more of the trials, more patients?
Right. So I know a bit about this, but not all that much. The trial now is limited to people, I think they have to be over 25. I've forgotten that, 25 years of age. But more importantly, as you said, George, they have to have had at least 2 severe hypoglycemic events in the years before. They have to have largely uncontrollable blood sugars, have a high hemoglobin A1C. And I think you asked me, what do I think about that? I think that's a very restrictive criteria. I'm told that in the United States, there are maybe 40,000 people who might fall into that out of the sort of 2 million type 1 diabetics. So it does restrict access. Another number I know, which I don't think is confidential, is after there was Vertex's press release, I think I heard that 15,000 people went to their website to try to sign up for the clinical trial. So I think I understand why the FDA and its equivalent in Canada is conservative about these things, but I'm hopeful as we move forward that one can reconsider the risk reward of taking immunosuppressants versus the daily monitoring and psychosocial costs of dealing with diabetes. Neither of those is a good situation. The question is, which would a patient like? So I'm not sure I answered the question, but you are absolutely right. It's very restrictive. It's not, children are not eligible. And you have to have had what I think used to be called brittle diabetes, but I don't think that term is used anymore.
Yeah. Yes. Okay. Thanks. Another question relates from Steve Grover, typical survival of islet producing stem cells in humans. Any long-term results? How long are we there so far?
Well, I think so far we're only 9 months in, maybe close to a year, but there's not even any direct evidence for survival.
Yeah.
All I can say is the half of the calculated dose was put in and it achieved blood glucose control. The patient is still well controlled, but we don't actually know. If you want my intuition, it would be this. What we know is based on using cadaveric islets. And remember, those were isolated from a deceased person by enzymatic digestion and then shipped somewhere. So they were hardly like a gold standard islet. We're making the equivalent of newborn baby islets, which I think might last a long time. You notice I'm not answering the question. We don't know how long they'll last, but my intuition is they're going to be very long-lived.
Could I ask a question about the differentiation?
Sure.
I was reading about that you put the cells in just before they're actually the perfect mature beta cell, and in vivo, they certainly in mice, they then differentiate to the final stages. Is there something that's still unknown about those last stages?
Well, there are a couple of, the literature is a bit confusing because there are two different kinds of cells being used. The company ViaCyte in San Diego uses progenitor cells, which mature into some beta cells after 4 months in a mouse or a year in a human. They did a very good clinical trial, but unfortunately, most of the cells died. And of those that did differentiate, most became alpha cells, not beta cells. And so they weren't able to achieve a clinically or therapeutically relevant dose. The cells I'm talking about today, George, and that Vertex used are already functional. They respond to glucose and secrete insulin. What happens post-transplantation is they get better and better. They ramp up their insulin production. So in that sense, they are mature. But I often joke, maybe maturation was a bad term and I'm responsible for using it. It's like, I could ask you, are you mature? And if you were mature, what year did you become mature? I mean, once they get vascularized, they just form better and better, but they're already fully functional when they go in.
We have one more question here about taking cells from a placental bank. Do you have any comments on would they be taken from placenta?
Yeah, that's another nice question. Thank you for the question. I get many emails from parents who've recently had a child and have frozen their umbilical cord cells, and then unfortunately some time later discover their child has diabetes. And they ask, is there any use of the umbilical cord cells? I can't think of a good use at the moment because, first of all, if the child has autoimmune disease, it's not going to do any good. It is true that the companies which sell the procedure of freezing these cells say that those cells can become any cell in the body. In fact, technically what they say is some people say they can become any cell in the body. It's true, some people say that, but no one's ever proven it. So I usually tell them, if you can afford to keep the cells frozen, do that for a while, but I can't think of any use of them at the moment.
We have two related questions here. We have a couple more minutes. The one relates to taking insulinoma cells that are relatively benign and see if those can be used. And the other relates to islet cells from ES cells. And do they divide after the transplantation? Are they capable of dividing?
Both great questions. So insulinoma cells do not accurately read blood sugar and secrete insulin. So they would be useful if you wanted to make them comparable to like a long, slow-acting insulin if you adjusted the dose. But they're not useful beyond that. They don't control blood sugar as well because they don't respond to glucose with pulsatile release of insulin. The second question was, what was the second question? I already forgot.
It relates to cell division.
Oh, right.
Okay.
So beta cells in a non-diabetic person, the beta cells you have in your body were pretty much there after you were a young person. They divide, as best as I can estimate from autopsies, about 8 times during your entire life. They divide very slowly. So on any given day, maybe 1,000 to 10,000 beta cells will be dividing in a normal human pancreas. So beta cells are like your brain cells, very, very long-lived. The cells we make similarly divide, but at a very slow rate. So as a bulk preparation, I would call them post-mitotic, but there are some dividing beta cells.
George, I think it's 1 o'clock, so we're going to have to end the session.
Can I just make one comment?
Please do, please do. Yeah.
The last chat here, we'll skip one question on immunosuppression, but just a point of advertising. Steve Paraskevas is thanking Doug and looking forward, since he's running the active site for Vertex here at the MUHC, we are accepting patients for this study.
Oh, that's great news. Thank you. I'm cheering you on.
Okay. Well, thank you very much, Doug. It was a fantastic lecture. I'm going to hand it back to Nadia. Thank you for rounds, and we'll see you this
Thank
afternoon.
you.
Thank you very much. I'm very thrilled to have had this special lecture today. Very inspirational, certainly, and transformative work. So thank you for that. And just a reminder to the crowd here today that there's going to be a second session at 5 p.m. today for those of you who want to hear more. I wish you the best of luck in your research, and I do hope that your dreams do come true, because they are very noble, and would be wonderful if they did. Could obviously be change many people's lives. So I wish you the best with your ongoing research, and thanks everyone for joining. Thanks, George, for
Thank you. Thanks,
for
Nadia.
helping to host today.
Thank
Have a great
you.
afternoon, everyone. Bye
Thank
bye.
you. Bye-bye.